All right, let's dive into a truly incredible breakthrough in cancer research. For decades, some of the absolute worst cancer-causing proteins have been hiding in plain sight, earning them this frustrating nickname, undruggable. But what if science finally figured out a way to rip off that invisibility cloak and take them down for good? Let's get into it. This isn't just some hypothetical question. For so many people, it's been the brutal reality of their fight. Imagine you've got this one villain. You know they're causing all the chaos, but every weapon you have just passes right through them. That is the exact maddening problem scientists have been up against with some of the most aggressive cancers out there. So who are these villains we're talking about? Well, the big names are proteins like MIC and KRAS. And we're not talking about minor players here. These are some of the most notorious drivers of cancer growth we know of. They are at the heart of a huge number of human cancers, and for decades they've been one step ahead of pretty much every treatment scientists have thrown at them. So you gotta ask, why? Why are they so hard to fight? Well, most drugs work kind of like a key fitting into a lock. They need a very specific, well-defined little pocket or groove on a protein that they can slot into and shut it down. But proteins like MYC and KRAS, they're different. They're often described as slippery, disordered. They just don't have those neat little pockets, making it almost impossible for a drug to get a solid grip. And this quote from the researchers just nails the frustration. It says finding a drug for these targets takes arduous optimization, which is scientists speak for a very, very long and difficult process. And even after all that work, it's pretty much useless unless you can find one of those rare ligandable pockets. It was so clear that to beat these proteins, science needed a totally new playbook.
And that is where our hero enters the story. Researchers at Northwestern University have engineered something completely different, a brilliant new strategy designed not just to block these villains, but to completely get rid of them.
They call them hydrax. Now, instead of trying to design a tiny key for a lock that doesn't exist, hydrax are a whole new kind of tool. Think of them as custom-built microscopic chains, or polymers, that basically go on a search and destroy mission inside the cell. Their only job is to hunt down that bad protein, grab a hold of it, and tag it for the cell's own garbage disposal system. And here's the really crucial part, the elegance of it. Remember that arduous optimization we just talked about? This new tech is a one-step polymer chemistry solution. It's a platform built from the ground up specifically to go after these slippery, disordered proteins that other drugs just can't touch. So how does it actually work? Well, the concept is so simple and effective, they call it the two-handed takedown. Just picture this. The hydrac polymer has two hands. With one hand, it reaches out and grabs our target, let's say it's the MYC protein. At the same time, with its other hand, it grabs the cell's trash can, which is its natural machinery for breaking down junk proteins. Then all it does is bring the two together. It physically forces the cell to see MYC as trash, and poof, it gets destroyed. And if you need a simpler way to think about it, the lead researcher, Nathan Giannoski, put it perfectly. He said it's literally like you see a piece of trash on the floor, you grab it, you grab the wastebasket, and you put them right next to each other. It's that simple. A physical solution to a really complex biological problem.
Okay, so the theory sounds amazing, right? But the big question is always, does it actually work? Well, the team put these hydrax to the test, and let me tell you, the results are seriously promising.
But first, let's just remember what's at stake here. One of their main targets, KRAS, is involved in roughly a quarter of all human cancers. We're talking about some of the toughest ones out there, like pancreatic and colorectal cancers. A technology that can actually take down KRAS is, well, it's a massive game changer. And the results from the lab were crystal clear. When they tested this on cancer cells in a dish, the hydrax did exactly what they were designed to do. They found and destroyed both MIC and KRAS. And this wasn't just some minor effect. It completely shut down the cancer-driving gene activity. And most importantly, it actually triggered the cancer cells to die. So the next big step, of course, was to see if it could work in a living system. And again, success. In mouse models, the hydrodeck polymers actually found their way to the tumors, they slowed down the cancer cell's ability to multiply, and ultimately, they stalled the tumor's growth. That is a huge proof of concept. Now, this is where the story gets even bigger, because this isn't just about finding one new drug for one target. The hydrax system is really a whole new way of thinking about how we design targeted treatments. So here's the million-dollar question for any new cancer therapy. We know cancers are sneaky. They evolve, they mutate to resist drugs. A traditional drug can stop working just because that little lock it fits into changes shape. So how does this new approach deal with that? This is, hands down, my favorite quote from the entire study. Because the Hydrac isn't looking for one tiny, perfect spot to bind to, a mutation doesn't really matter as much. The polymer is grabbing the whole protein. And as Dr. Janeski says, it doesn't matter if that protein mutates, it's still going into the bin. He says CRAS can be kicking and screaming all the way to the trash can. This could be a really powerful way to get around drug resistance. And the potential, believe it or not, goes way beyond cancer. By showing that this platform works against two completely different undruggable proteins, the researchers think it can be adapted for all sorts of things. Because these hard-to-target proteins, they're also at the root of diseases like Alzheimer's, Parkinson's, and many inflammatory or metabolic disorders. This could be the key that unlocks treatments for them too. So we end on this really exciting thought. We started with a problem that for decades seemed impossible. These villains that science just couldn't touch. And we've ended with a brand new kind of hero that doesn't just fight them, it literally takes out the trash. The question is no longer if we can target these proteins, but which ones we should go after next.